This is a plankton net. This is the working end. This is a 1/2 meter net. The net is actually very fine. It's 200 microns in size. We have a little bit one of the largest size, a 300 micron net. And we've got two different types of cod ends, which is the catch bottle that we use. We have a solid cod end, which goes on the back end of the net, which actually catches the plankton. And would you show your cod end on the screen? So the net gets stung in the wall, hung in the water this way, and suspended off of this piece or these wires. Normally, we put a meter in the ring like so we can measure the volume of water that goes through so we can actually compute plankton densities. We're not going to be doing that today because our main objective is identification, not quantification. So
we also have this type, which has mesh along the sides to let water out as well. It's 365 microns. Put that in relative context for you guys. An average hair is around 100 microns. So that's the width of your own hair. So this is really fine now.
go ahead and pull that down.
This one down.
They're almost like insects, but in the ocean. In fact, they're distantly related to insects. Copepods. Do you have them? Yeah, it looks like there's a hard shell in the middle, and then those are the wings on the side. Pteropod, that's beautiful. Boy, make sure.
My name is Nitzan Sofer, and I'm a graduate student at Florida International University. I study diseases in coral reefs in Dr. Vega Thurber's lab.
I'm Rory Welsh. I work for Florida International University and Dr. Rebecca Vega Thurber's lab.
We have just come back from the boat and have brought into the lab our samples. And so I'd like to for you two to tell me what's in them. Tell me something about the diversity, the biodiversity of what we can find in the plankton.
Okay. Well, we have two different size mesh nets that we're using. Here we have the 300 micron, and this is the 200 micron. So anything less than 200 microns probably isn't going to be contained in these samples. It's more the larger stuff. And these were just done on plankton toes, 20-minute toes, and we've got isopods, copepods, and all sorts of other different types of plankton, as well as some larval fish, like this one that Nitzan just identified as a juvenile puffer.
Oh, super. Got your sample there.
Okay, so we have two kinds of plankton. We have zooplankton and phytoplankton. Now, zooplankton are animals like shrimps and copepods and different crustaceans. And the phytoplankton are actually plant material like diatoms or dinoflagellates. And plankton and zooplankton are usually very small that are on the surface. And the kind of fish that you might find swimming on the surface are usually larvae, which are baby fish. And the larvae, we have all sorts of types, and even here we found puffer fish. And earlier we found a larvae that actually had a yolk attached to it, which means that it's very fresh, maybe just a couple days.
Oh, you can find a wide variety of animals in the samples that we collect. We've got everything from the phytoplankton, which are like plants. The most abundant organism on the planet, like Chlorococcus, should be in here, although they're mainly found in open oceans, and their numbers and abundance decrease towards coastal waters, whereas their counterpart in the cyanobacteria, the Synechococcus, take over. And then the much larger stuff, we have jellies, we have arthropods, isopods, copepods. It's just a wide range, and we're just getting a glimpse at the biodiversity that's out there.
Thank you very much indeed, guys.